Good evening, one and all. So today we are going to discuss about our new topic, which is nutrition. Here we are going to cover two topics in this video, which involves holozoic nutrition first. What is nutrition from basic, and what is the mechanism of nutrition in amoeba? What is the mechanism of uh, nutrition in a unicellular eukaryote known as amoeba? So we are going to first start with uh, the very basic. What is nutrients? So here, what are nutrients? Nutrients are essential food substances that are acquired for metabolic activities. All of the living organisms in this world require food in order to grow, in order to repair, in order to maintain their metabolic life activities, in order to sustain themselves. So nutrients are essential food substances that are acquired for metabolic activities. What is meant by essential here? Essential means that something which is important but not synthesized in our body. Something that is important for our body but not synthesized in our body. Non-essential on the other hand side means there's something which is also important but it is synthesized in our body. As we are having essential and non-essential amino acids, there's the same case with essential and non-essential here. So essential food substances that are acquired for metabolic activities, growth, repairment, as well as for sustaining life and other such stuff. Afterwards, we are having nutrition. So nutrition is the process of intake of nutrients for growth, for repairing of materials, as well as for movement, locomotion, cell division. All involves nutrients. All involves in the process of nutrition. Afterwards, we are having the types of nutrition, in which we are going to discuss about a few things. First, we are going to discuss about what is nutrition. Nutrition is the process of intake of nutrients for growth, repairing, movement, locomotion, cell division. Afterwards, we are having autotrophic nutrition. Autotrophic nutrition here means you know, nutrition in organisms, nutrition in organisms which can produce their own food by converting inorganic food, inorganic molecules into organic molecules, by converting inorganic molecules into organic molecules, inorganic molecules into organic molecules through process of photosynthesis, either process of photosynthesis or chemosynthesis. So here there are two processes which are involved in creation of food by organisms by converting inorganic substances into organic substances, the photosynthesis or chemosynthesis. This process is known as autotrophic nutrition, which is acquired by some type of living organism such as plants, or we are going to have a good example of some bacteria, photosynthetic and chemosynthetic bacteria, as well as we are having algae, which has a good example, autotrophic nutrition. Afterwards, let us discuss about. So in this case, we are having autotrophic nutrition, and organisms undergo photosynthesis or chemosynthesis for producing their own food using autotrophic nutrition. So they have the capability of producing their food by converting inorganic into organic molecules. And a good type of autotrophic nutrition is saprophytic nutrition. Saprophytic nutrition involves organisms who can involves organisms who feed on dead animals, plants, or other matter, which feeds on dead animals, plants, or other organisms. These types of autotrophic, these type of saprophytic nutrition involves decomposers as a good example, as there are many decomposers which decompose over the dead material of animals, plants, organisms, or other organisms, and these basically work as decomposers in order to sustain themselves by producing their own food. They actually feed on actually feed on dead animals and plants, but they produce their own food by converting the inorganic molecules into inorganic molecules. This time, the saprophytic nutrition convert organic to inorganic molecules. So suppose if a molecule of protein is there, so they would be converting that molecule of protein into amino acid and then amino acid into inorganic molecule, and then it would be getting energy from that. Afterwards, we have heterotrophic nutrition. Heterotrophic nutrition involves organisms which are incapable of producing their own food, due to which they obtain food from autotrophs. They are incapable of producing their own food from converting inorganic into organic substances, due to which they depend on autotrophs for their nutrition. And heterotrophic nutrition involves fungi, and animals are examples of heterotrophic nutrition. There are types of heterotrophic nutrition, including detrivore. What is detrivore? Detrivore are organisms which feed on detritus. Now, what is detritus? Detritus uh, is, in simple words, organic debris. Organic debris is known as detritus. Now, what is debris here? So, debris is decomposed matter. Now, organic decomposed matter is known as detritus, and detritus is fed by detrivores. Afterwards, we are having fluid feeders. Fluid feeders are those heterotrophic organisms which feed on the fluid of other organisms. Fluid Feed on fluid of organisms. Example of this can be mosquito. Example of this can be mosquito. Afterwards, we are having filter feeders. Filter feeders are those organisms which filter organic molecules from water, and by filtering those organic molecules, they utilize those organic molecules as a source of nutrition. As they utilize those organic molecules as a source of nutrition, these organisms are considered as filter feeders. Examples of this can be mussels. Afterwards, we are having holozoic nutrition. At last, what is holozoic nutrition? Word holo means entire or whole, whereas zoic refers to animals. Zoic refers to animals. So holozoic means holozoic nutrition is a type of nutrition acquired by heterotrophic organisms who intake complex food molecules by their lumen or cavity in order to digest them. So they intake complex food molecules from autotrophs inside their lumen or body cavity in order to digest them. So holozoic nutrition involves organisms which intake complex food molecules inside their body in order to digest those complex food molecules inside their body. They digest those complex food molecules inside their body. And how do they intake that particular complex food molecules? That complex food molecule is intaken by a lumen or cavity inside that organism's body. So that is how holozoic nutrition gain their nutrition. So when it comes to nutrition, the nutrition is of two types: macromolecules and micromolecules. As we have previously discussed, what is macromolecules and macromolecules? Macromolecules are those which are having complex structure. These are indiffusible. These are having complex structure. These are larger molecules. These are polymers. These are basically having higher molecular weight, as well as larger molecules and other characteristics of macromolecules as well. When it comes to micromolecules, these involves smaller molecules. These are smaller molecules. These are simple molecules. These are diffusible molecules, as well as these are the monomers, and these are having low molecular weight. Here, some macromolecules involves carbohydrates. Micromolecules involves glucose of the carbohydrate. After which we are having protein. Protein is having its macromolecule as amino acid. Amino acid. After which we are having the lipids. Lipids are having its micromolecules as fatty acids and glycerol. Fatty acids and glycerol. After which we are having nucleic acid. Nucleic acid is having its micromolecule as nucleotide. So these are this is a basic definition, basic description about micromolecules and macromolecules. After which we are going to discuss about ways of getting nutrition in organisms. There are in holozoic organisms there are the following ways of getting nutrition. First step involves ingestion. What is ingestion? Ingestion is a process in which organisms intake complex food molecules through a lumen or cavity, which is allowing the complex food molecules to be taken inside the organism. So ingestion involves taking in of food. For instance, we are having ingestion is taking in of food of complex food molecules, and these are taken through a lumen or cavity of body. And in humans, that lumen or cavity is known as buccal cavity, through which we ingest the food, and after ingesting, the digestion of the food takes place. Now, digestion of the food is of two types. First of all, let us discuss what is digestion. Digestion is a process in which breakdown of large, complex, the indiffusible food molecules takes place into diffusible, smaller, simpler micromolecules in order to get diffused into the particular cell of that living organism. For instance, we are having the digestion of carbohydrates, proteins, lipids inside the human body. These are all examples of digestion, and digestion here involves the conversion of macromolecules into micromolecules. That is of two types: chemical digestion and mechanical digestion. Mechanical digestion is digestion which takes place in mouth and inside the stomach. In mouth, we are having the teeth. In mouth, we are having teeth, and teeth are having what? Teeth are able to chew the food. Through teeth, we are able to chew the food, and this process of chewing the food through our teeth is an example of physical digestion, and that is in biological terminology named as mastication. On the other hand side, in stomach, we are having circular muscles and oblique muscles. And oblique muscles, Circular and oblique muscles, which are basically, these are circular and oblique muscles which are assisting in the physical digestion, mechanical digestion of food. These help in breakdown of food, breakdown of breakdown of food into smaller molecules. In mechanical digestion, there is no breakdown of chemical bonds. There is only the breakdown of large food molecules into smaller ones. Afterwards, we are having chemical digestion. Chemical digestion involves the digestion through the breakdown of chemical bonds. That involves the use of biocatalysts, which are enzymes, as well as several digestive juices present inside the alimentary canal of humans. So here we have a good example of the salivary amylase. In mouse, we are having salivary amylase. In mouse, we are having salivary amylase, which is able to convert starch into maltose. So it is able to convert starch into maltose, and 30% of the uh, maltose here, 30% of starch is digested in mouth through chemical digestion. 30% of starch is digested in mouth through chemical digestion, and that is a good example of chemical digestion. In stomach, we are having several enzymes, digestive enzymes such as pepsin, as well as lipase, and there are other examples as well. When it comes to the small intestine, inside small intestine, we are having several juices such as pancreatic juice, bile juice, HCl. All are examples of some juices inside the alimentary canal as well. So these are all involved in chemical digestion for conversion of food from macromolecules into micromolecules. After which we are having absorption. Here, absorption refers to the intake of diffusible food molecules, intake of diffusible food molecules into the cell. Intake of diffusible food molecules, digested food molecules inside the cell, which takes place with the help of absorption. This process is known as absorption, and this occurs uh, by means of bloodstream, by means of bloodstream, lymphatic system, which actually opens inside the bloodstream through interjugular vein, and it can directly be absorbed inside the cells as well. Cells such as cells of in a small intestine directly absorb the diffusible food molecules. Afterwards, if the food is digested, if there's digested food that would be absorbed through these means, that would be absorbed through these three pathways, which is lymphatic system, bloodstream, or cell itself directly. Or if it is undigested food, if it is undigested food, where it will go? If it is undigested food, it will go inside large intestine, go inside large intestine, where further what will happen further, the absorption will take place of water molecules as well as some minerals which are important for our body. So that food will be that important material will be absorbed from the food inside the large intestine, the particles inside the large intestine actually. Assimilation is the utilization of that particular food which is absorbed inside the cells. That utilization is for what? Utilization involves glucose first. Glucose is utilized for cellular respiration to create what? It is for creating energy, ATP. It is for creating ATP. After which we are having the amino acids. Amino acids are utilized for creating what? Amino acids are utilized for protein synthesis inside the ribosomes. After which we are having fatty acids. Fatty acids are used for insulation, shape, protection, and reservation of energy inside the cell. So fatty acids are utilized for several purposes: reservation of energy, shape, protection, as well as insulation of cell. And these are also found in the adipose tissues, where these act as insulation as well as protect the body, as well as these also act as a reservation of energy, and these are also providing insulation to the body as well. Afterwards, when it comes to undigested part, undigested food which is present in large intestine will be reabsorbed. Some of the material which is important will be reabsorbed, after which the final step is egestion. Egestion is a process in which removal of unwanted food materials will take place. Removal of undigested particles will take place from the body. Undigested particles, which involves what? Undigested particles such as cellulose, rough phage fibers would be eradicated from the body, as well as nitrogenous compounds would be eradicated from the body, from the large intestine into rectum, and from rectum into the outside of body. And cellulose is also taken out from our body without digestion. It is important for our body in order to allow the smooth movement of undigested particles from the body, and that is required in order to prevent conditions such as constipation. After which we are having types of digestion. Types of digestion involves intracellular digestion and extracellular or intracellular digestion. Intracellular digestion is a digestion in which the digestion of food particles takes place inside the cell, as well as the enzymes which are produced in order to the enzymes which are produced in order to digest those food particles are also produced inside the cell. Enzymes are also produced inside the cell. After which, when it comes to extracellular digestion or intracellular digestion is a digestion outside the cell. Digestion outside the cell, and the enzymes are also produced outside the cell. Enzymes outside the cell are produced. After which we are having so The intracellular digestion is divided into two organisms. It occurs in two organisms: unicellular eukaryotes, multicellular eukaryotes. In unicellular eukaryotes, we are having examples of Amoeba paramecium. In multicellular eukaryotes, we are having Hydra planaria and some of the insects which are specific. After which, we are having intracellular, extracellular digestion, in which digestion occurs outside the cell, and that type of digestion takes place in higher living organisms. By the way, in higher living organisms, both type of digestion takes place in higher living organisms. Extracellular digestion takes place as well as intracellular digestion takes place as well. So now let us study about the digestion, the process of nutrition in amoeba. Here in amoeba, what actually happens is that in amoeba we have Golgi body, and that Golgi body produces some hydrolytic enzymes which are known as lysosomal enzymes. Some lysosomal hydrolytic enzymes by budding off. How does it produces the hydrolytic lysosomal enzymes? By budding off a membrane, from its trans Golgi network. By budding of a membrane from its trans Golgi network, but budding of a network from its trans Golgi network, it produces a lysosomal enzyme. By budding of a membrane from its trans Golgi network, it produces hydrolytic enzymes and lysosomal enzymes. And then by budding of a membrane from its trans Golgi network, it produces a membrane around that hydrolytic enzyme, causing to form an organelle known as lysosome. That causes to form lysosome, and that lysosome is basically utilized for several type of organelles. It is used for digestion of food molecules because it contains the hydrolytic enzyme. As it contains hydrolytic enzymes, these hydrolytic enzymes are supposed to help in the breakdown of food molecules from macro into micromolecules. And what is the pH of these hydrolytic enzymes? The pH of these hydrolytic enzymes is 5.6 to 7.3. After which we are having after which we are having the steps of digestion here again. So as we know that amoeba contains feet-like tentacles or projections on its pseudopodia. Now in this case, what actually happens whenever a food particle comes near the cell, so the cell starts to the pseudopodia starts to grasp that food particle. The pseudopodia starts to grasp the food particle. As you can see in the second part, the pseudopodia starts to grasp this food particle due to the result of chemotactic movement. Here, chemotactic movement takes place due to which the pseudopodia comes near the food particle and grasps it. As it grasps it, the cell membrane folds itself. The cell membrane folds itself and stretches and folds itself, due to which the food particle is covered inside the cell membrane, due to which happens in the next step. Endocytosis takes place. Now endocytosis is of two types: phagocytosis and pinocytosis. Pinocytosis is the intake of water. Pinocytosis is the intake of liquid inside the cell, whereas phagocytosis is the intake of solid particles, food particles inside the cell. So phagocytosis takes place, in which what happens, the food particles are grasped by pseudopodia, and after grasping, the cell membrane folds around the food particle, due to which it stretches and folds around the food particle, and that food particle has the membrane enfolded outside it. So the cell membrane causes to form a membrane outside the food particle, and this membrane which is formed outside the food particle is known as unit membrane. And after this formation of unit membrane around the food particle, that food particle will be converted into a food vacuole. It is converted inside a food vacuole. As it is converted into a food vacuole, that food vacuole is also known as phagosome. That food vacuole is also known as phagosome. That food vacuole is also known as phagosome. So this food particle, this food vacuole is known as phagosome. This food vacuole is known as phagosome. So here, as we can see that after the formation of phagosome, that phagosome is carried. So what happens here is that the food vacuole, after the formation of food vacuole, the food vacuole is carried towards the lysosome by the process of cyclosis. Cyclosis is the movement of cytoplasm, due to which the food vacuole is carried towards the lysosome, and that lysosome fuses with the food vacuole. As lysosome fuses with food vacuoles, the enzymes of lysosome, lysosomal enzyme, hydrolytic enzymes, act over the food particle, due to which the food particle breaks down from complex to simpler food molecules, which means it converts from macromolecules into micromolecules, from polymers into monomers. After which this process leads into completion of digestion, and the pH of all of the particular enzymes which are present inside the lysosome depends on the food particle, depends on the food component which is being digested. So the pH of this lysosomal enzymes depends on the food component which is having these food particles digested. So after the digestion, what will happen? Of course, absorption will take place. As absorption will take place, that absorption will take place by bringing those digested macromolecules outside the food vacuole, outside the lysosome. Due to which, what will happen after digestion? The particles will come outside food vacuole, and through cyclosis. Cyclosis will allow the movement of cytoplasm. That allows movement of cytoplasm. Through cyclosis, the food particles will be transported to the respective parts of the cell, to respective organelles wherever it is required. For suppose if a cell requires carbohydrates, then if a cell organelle requires carbohydrates, then it would be transported towards towards the particular cell organelle mitochondria. Through the process of cyclosis, cyclosis allows the movement of cell. As the cyclosis allows the movement of cytoplasm, and alongside cytoplasm, the cell organelles as well. After which we are having what? After which we are having our next step, which is assimilation. Assimilation involves the usage of those food particles. For instance, in mitochondria, the glucose will be used for cellular respiration to produce energy, ATP. After which, for fat droplets inside the amoeba structure, there are fat droplets which are produced by fatty acids. So fatty acids is combined together in order to produce fat droplets, and these fat droplets will be utilized for the insulation of the amoebic cell, as well as in order to provide stabilization to the amoebic cell by acting as a buffer in the amoebic cell as well. When it comes to protein, protein will be having its monomer as amino acids. Amino acid will be utilized in protein synthesis in ribosome, due to which other metabolic functions, other metabolic activities can be done inside the cell organelle. After which there would be after assimilation, we would be having egestion finally. In egestion, what we would be having? Egestion, we would be having unwanted particles. There would be undigested particles. What would be those undigested particles? There would be H2O, there would be carbon dioxide, excessive H2O. H2O which is excessive would be ingested, as well as carbon dioxide and ammonia. Afterwards, all of these three compounds will be exited outside the cell through exocytosis. And exocytosis involves what? First, what will happen? Unit membrane will be formed outside these particular undigested particles. As the unit membrane will be formed outside these particular undigested particles, these undigested particles will be carried near the surface of cell membrane. As they would be carried towards the surface of cell membrane, the unit membrane of those secretory vesicle, actually these particles, when we will be forming the unit membrane, will be called as secretory vesicle. So they will be forming secretory vesicle, and when these secretory vesicle will be going on the surface of cell membrane, these secretory vesicle uh, the secretory vesicles unit membrane will fuse with the cell membrane, due to which the undigested particle will come out of the cell, from the cell membrane, and go out of the cell. So this is the process in which the nutrition of amoeba takes place. The amoeba itself digests and gains nutrients from its surrounding food particles. Hopefully you enjoyed the video, and do share the video if you have any questions.